Alright guys, so this video is going to cover cells. So we're starting our new unit, and typically students are going to find this unit a little bit easier than the last one because cells are just going to be more familiar than biochemistry. And so it's actually a relatively short set of notes. So looking ahead to the quiz, you will have a quiz on both this set of notes as well as the cell parts and organelle lab that you're going to be doing for the next couple of days. So you're going to be expected to know all of the content of this set of notes, which is pretty much going to be cell theory, the two main types of cells, some microscopes, and a little bit of evolutionary content. And then the other set of notes, which will be labeling the parts of the cell as well as knowing the parts and what they do. And so just starting off, we have the cell theory. And so this was actually put together from the work of three main people. We have Schleiden, Virchow, and Schwann. And you're going to be getting a bonus today, and that bonus will have these people's names on it. And so you'll kind of get an idea of what each person did specifically. You don't have to know that for the quiz or the test or anything like that. But the cell theory essentially has three components. And one thing you have to understand is that at some point, somebody had to draw this conclusion, okay? It may seem kind of silly to us today to say that all living things are composed of cells. It's kind of like a, well, duh. But at some point, people had to make these conclusions. And so Schleiden was a botanist, and he studied plants. Schwann was a zoologist. He studied animals, and they both made conclusions like all plants are composed of cells and all animals are composed of cells. You know, keep in mind there was a time when we couldn't even see cells. And so the first statement is just simply that all living things, and this is important, these three statements you should just know, but there have been times where people will say all things or everything, and that's not the case. It's all living things are composed of one or more cells. The second one is cells are the basic unit of structure and function. Sometimes this can be simple, more simply put as cells are the basic unit of life. Either one is acceptable. Okay, so cells are the basic unit of structure and function. Remember, in our organization, cells are where life begins. And then the last one is cells come from pre-existing cells. And this comes from the work of Virchow, and I believe there's some other people that maybe made some similar conclusions. But once again, there was a time when people believed that life could arise from non-life, that microorganisms could arise from, the earth, soil, gravy. There were some experiments that were done to kind of reject the idea that life could come from non-living matter. And so today we know that cells come from other cells. Okay, life comes from life. So once again, you just put the three statements together. You do just need to know these statements, and they are all living things are composed of one or more cells. Cells are the basic unit of structure and function, and cells come from pre-existing cells. All right, now cells are tiny. We know this. Cells are microscopic. Typically, we have to use a microscope to see them, but they are small for a reason, okay? And a lot of times this can be confusing to students, but cells must be small in order to maintain a large surface area to volume ratio, okay? And so the thing is big cells become inefficient. And so if we kind of look at the math down here, okay? So a couple of things. We have surface area and we have volume. I'm going to try to switch over here to a pen. I don't know how well that is going to go. But surface area is measured in units squared. Okay, and surface area is going to be the area of the surface. Now we have cubes here just because they're easy to calculate, but of course cells are going to be irregular. And so one thing I want you to remember is that the surface area of a cell is going to be the cell membrane. And the cell membrane's job is to regulate what enters and exits the cell. We're going to have a whole lesson on that. It's also going to be on your cell parts lab. And so the surface area is around the outside of that cell. That's going to be your cell membrane. and its job is to be the gatekeeper. Now, the volume is going to be everything else, and volume is measured with cube units, okay? And so the volume is going to be all the inside of a cell. It's going to be the organelles, it's going to be the cytoplasm, it's going to be essentially everything. And so one thing I need you to understand is just some basic math, okay? So if this cell gets bigger and bigger and bigger, like we see in the picture, okay, both the surface area and the volume are going to increase. However, they are not going to increase at the same rate. So since surface area is a squared unit and volume is a cubed unit, the volume is going to increase more than the surface area will. And so once again, that cell membrane's job is to regulate the in and out. And so the bigger that cell gets, the more that cell membrane is going to have to do. But yet there's more internal volume than there is surface area, okay? So cells must be tiny to maintain a large surface area to volume ratio. So here if we have a measurement of, let's say, 1 centimeter, okay, the surface area is 6, the volume is 1, and so that gives us a ratio of 6. Okay, now if we double the side measurement, we now have a surface area of 24 and a volume of 8. So notice that that ratio is going down. Okay, that means there's not as much outside per inside. We get even bigger. Once again, we see that ratio decreasing again. So that means there's more internal components than there are to that external membrane. So even though both grow, the volume grows faster. And so the smaller the cell, the more efficient it's going to be, because that cell membrane can get bogged down if the cell gets too large. Also keep in mind that the DNA is the, kind of the regulator of the cell. There's not more DNA in a big cell. And so in order to keep up with all of the cell's activities, cells must be tiny. And so here's just kind of a basic size comparison. We've got, atoms down here, which remember they make up everything. We have our macromolecules. Small molecules will be something like water. Your macromolecules are going to be, quite a bit larger. Nanometers are billionths of meters, just to kind of give you an idea. This symbol here is micrometer or micrometer. Those are millionths of meters, and so we can kind of see the basic size comparison. So atoms are incredibly tiny. Obviously, they make up everything. Here we have some of our bigger molecules. Here we have our small organelles. When we get up here, we have larger organelles. And so cells kind of begin in this micrometer range. Most of your typical plant and animal cells are going to be in this micrometer range here. And so we can use light microscopes to see these items. In order to see these items, we're going to have electron microscopes. And so just a little bit about microscopes. Now, you will be using microscopes throughout the course, but typically I'll already have them magnified for you. I hope you remember from freshman biology. But we do have two main types of microscopes that we're going to look at. We have light microscopes, which obviously, stating the obvious, they use light in order to create an image. Now, typically your light microscopes can go up to about 1,000 times. They can allow us to see bacteria and things like that, but even bacteria are going to be hard to see well with the light microscope. One of the benefits of a light microscope is you can look at live specimens. So if you wanted to put pond water on there, you could see little microorganisms moving around. But one of the downsides is it does have to be very thin, and it has to be somewhat translucent. Otherwise, you're just going to see a shadow. So it's really going to be difficult to see like whole organisms, even if they're small, because unless they're translucent, all we're going to see is a dark spot. So it has to be thin, which is often why we look at things like cross sections in order to look at images under the light microscope. Now, the best microscopes are the electron microscopes, and so they are going to use electrons to produce an image. The only downside is that you do have to have a specimen that is preserved. You cannot look at live specimen with an electron microscope. However, the pictures are much, much, much better and more detailed. That's because they magnify about 1,000 times more than a light microscope. So we're talking about 1,000,000 times magnification there. Now there are two types. You have a transmission electron microscope, and in a transmission electron microscope image, the electrons are passing through, and so we use this to kind of study infrastructure. If you wanted to look inside of a cell, if you wanted to study the structure of a mitochondria, something like that, you're going to want to use a transmission electron microscope. And this picture up here is your transmission electron microscope, or TEM. And notice we can see the internal components. Now, if you want to produce really cool images, that's when you're going to use a scanning electron microscope. The electrons are actually kind of used to scan the surface, and this is where we get 3D images. So up here you see the spider, so that's a scanning electron microscope. These are probably pollen grains, that's a scanning electron microscope. And you can see that they give you a very detailed 3D type of image. And so our best pictures, unlike a lot of the pictures that you're going to see in textbooks, things of that nature, are going to be created using electron microscopes. You will kind of need to know the differences about those for the quiz. All right, so now let's talk about cells themselves, okay? Now, all cells, and this is kind of a bold statement, but all cells are going to have 3 basic features. And I'm actually going to have you add one because I've never seen an exception. So we're going to say actually that all cells have 4 basic features. Number 1 is all cells have a plasma membrane around the outside, also known as the cell membrane, that gatekeeper that regulates the in and out of the cell. Number 2, all cells contain genetic material, and we're talking about DNA here, okay? Also, all cells are going to have RNA, so technically both. All right, so all cells have genetic material, and then all cells are going to have cytoplasm, which is the filler. It's going to be the space. The cytosol is the liquid within the cytoplasm. And so all cells are going to have these components. Now, there is one that I want you to add. Okay, I want you to add a 4th one, and that is ribosomes. Ribosomes are your protein factories, and you can find them in all cell types. This is where I'm missing my whiteboard right about now. Sorry about the messy writing. And so all cells, this is going to be important, no matter what type it is, no matter whether it's a prokaryote or a eukaryote, a plant, animal, fungus, all cells are going to have a cell membrane on the outside. They're going to have genetic material that kind of regulates or provides instruction. They're going to have cytoplasm, which is the filler, and they're going to have ribosomes, which make proteins. Now, aside from that, we can actually put cells into 2 main groups. Cells can be classified as either prokaryotes or eukaryotes. And so I'm really big on breaking down words, and so karyo refers to nucleus. And so if we take this word here, prokaryote, pro means before. So literally prokaryotes are before the nucleus. So they do not have nuclei, which just simply means that their genetic material is going to be without a home. The DNA is going to be kind of in just a region in the cytoplasm rather than in an actual home or membrane-bound structure like a nucleus. Now, eukaryote, eu means true. So this word literally means true nucleus. So we have before the nucleus and we have true nucleus. Now, prokaryotes actually fall in 2 domains, okay, of life. There's archaea, which are kind of extremophiles, and then there's bacteria, which are going to be your common everyday bacteria. And so prokaryotes are going to include those cells that fit into those 2 domains. And then eukaryotes have their own domain. It's domain eukarya, and that's going to include everything else, which are fungi, protists. Protists are things like amoebas, paramecia. A lot of them are unicellular. Some of them are big, but most of them are tiny. And then you have plants and animals. And so we're going to take a look at the key similarities and differences between these 2 types of cells. You're definitely going to want to understand how they are alike, but also how they are different for quiz purposes. And so in the picture here, we can see that the prokaryotic cell definitely kind of looks a little bit different, but there are some key things they have in common. There's that plasma membrane that all cells have, the cytoplasm that all cells have, the genetic material that all cells have, and the ribosomes that all cells have. And so we're going to start with the things they have in common. It's definitely going to be those 3 things that we mentioned on our list, or 4 things. They have genetic material. They have a cell membrane around the outside. They have ribosomes for producing proteins. They're also going to have cytoplasm. Now, they also have the same basic macromolecule composition. So we've already talked about carbs and lipids and proteins and nucleic acids. They're going to have the same types of macromolecules, the same basic structure. Remember that, organic compounds contain carbon. They make up all life, whether it's a prokaryotic cell or a eukaryotic cell. They also have similar metabolism in terms of how they make energy and do chemical reactions. And both groups have a wide variety of cells within them, so they're both very diverse. So we're going to start off by looking at the prokaryotes and see how they differ from the eukaryotes. And so remember that the key is prono. Okay, prono. Prokaryotes do not have a nucleus. And so since the DNA is found in the nucleus in a eukaryote, in a prokaryote, remember the DNA does not have a home. Instead, it's in what we call the nucleoid region. It's just the area where the DNA can be found. It's going to be within the cytoplasm. Now, these are believed to be the first cells in terms of an evolutionary perspective. Prokaryotes were the first cells, and they were believed to have evolved about maybe 3.5 billion years ago or so. Also, when we say prono, they do not have internal membrane structures. Okay. Now, it doesn't mean they can't have any organelles. They don't have membrane organelles, okay? They don't have internal membranous structures. They are smaller. A lot of times we say that they are simpler because they don't have quite as much stuff. They're about 1/10 of the size of a eukaryote. For those that do have a cell wall, which is actually going to be domain bacteria, inside of their cell wall they have a substance called peptidoglycan. And when it comes to classification, this is one of your identifiers. So your archaeans don't have this, but your bacteria groups do have peptidoglycan. And that's just a kind of a protein carbohydrate substance that you'll find in the cell wall. They can have flagella for moving, but it is a simpler structure. It's not surrounded by the plasma membrane. When it comes to chromosome structures, they have one, and it's a circle. Okay, whereas eukaryotes have these X-shaped chromosomes, and they have many. They may contain these extra small pieces of DNA called plasmids, and I'll show you a picture of that here in a second. And when they reproduce, they are asexual. They reproduce by a process called binary fission. It's essentially copying their DNA and splitting in two. The key to asexual is you don't really get variation. All right, so your eukaryotes are going to be the opposite of everything we just said. So you do. So we're pro-no, you do. They do have a nucleus, and that's where you'll find their DNA. They do appear to be more complex because they have more structures internally. They do have the membrane-bound organelles, things like mitochondria and chloroplasts, lysosomes. As you do your lab over the next couple of days, if the structures are membrane-bound, lysosomes, mitochondria, chloroplast, Golgi bodies, endoplasmic reticulum. If they're membrane-bound, they're going to be found in eukaryotes only. If they do not have a membrane, they may be found in both. Okay, so these are believed to have come after prokaryotes, about 1 billion years after. They're about 10 times larger. If they have a cell wall, now not all eukaryotes have a cell wall. Animal cells do not have one. But if they do have a cell wall, plants have cellulose and fungi have chitin. We actually looked at those two terms when we covered carbohydrates. Their chromosome structure is different, and they have multiple, and they are linear. And typically we just describe chromosomes as having an X shape. So where bacteria have one circular chromosome, eukaryotes have many X-shaped chromosomes. And when it comes to reproduction, some eukaryotes reproduce asexually. A lot of them reproduce sexually, which is going to involve creating variation in two parents. And so I just want to go back to the pictures here. Okay, so you can see there's a lot more going on in the eukaryotic cell, a lot more structures. Now, they do have those 4 things in common that we talked about. They both could have things like flagella. They both could have cell walls, but even in those, there could be some differences. So here we have the DNA kind of floating in the nucleoid region. Over here we have the DNA housed inside of the nucleus. All right, so just a little bit of evolutionary perspective. Okay, if prokaryotes came first and eukaryotes came second, how did that happen? Well, the most widely accepted concept of how we got eukaryotic cells is known as the endosymbiotic theory. Okay, now let's break this word down for a minute. So endo means in or inside of. Symbiotic refers to symbiosis, which is where two organisms are in a close relationship, and at least one is benefiting. So you may remember the terms mutualism, commensalism, parasitism, but endosymbiotic theory is the idea that prokaryotes, back when it was only a prokaryotic world, prokaryotes engulfed or took in other prokaryotic cells. And instead of digesting them for food, they actually lived together in a symbiotic relationship. We still see this today. We have bacteria that live inside of our intestines, and we have a symbiotic relationship with them. So they are alive and functioning. They help us, we help them. So the idea is that prokaryotes engulfed other prokaryotes, and the smaller one lived within the larger one. And then over time, these evolved into our modern day eukaryotic cells. The key evidence for this theory are these special organelles called chloroplast and mitochondria. They are literally like little bacteria. They are about the same size as a prokaryotic cell. They have a double membrane structure which supports that engulfing concept. They're a membrane surrounded by another membrane. They have their own DNA, and their DNA is a circular chromosome like that of a prokaryote. They have their own ribosomes. They can replicate themselves independently within our cells. Now, today, if you were to take a chloroplast or a mitochondria out of a cell, it would not be able to live independently because it's dependent upon the cell. And so that relationship, that symbiotic relationship, is what led to these organelles within. So chloroplasts are believed to have some photosynthetic ancestors. Mitochondria are believed to have some cell respirating ancestors. And so essentially these organelles are believed to be descendants of these internal prokaryotes that were engulfed. And so it's because of that special structure that they have. And so we'll talk more about this when we get to our actual evolution unit, but just remember that all things in biology can be answered through evolution. Even if you don't necessarily agree with it, you just have to understand it. All right, so just kind of some important dates. Life was believed to have originated about 4.5 billion years ago, with the first cells arising about 1 billion years after that. It's not until photosynthesis occurs that we get an oxygen-rich atmosphere that's going to allow for better energy production. And eukaryotes were believed to have come about 1.5 billion years ago. And so just a kind of a quick explanation of the endosymbiotic theory. So you had a larger prokaryote engulfing a smaller prokaryote, and over time, because of the symbiotic relationship, they kind of keep that these internal structures. And so once again, our evidence for this is going to be our mitochondria and our chloroplast. And so what would be the benefits of having these internal membrane structures? Well, they're going to help make things more efficient. They're going to help cell processes kind of have a home. Essentially, certain reactions can now take place within a specific compartment, and that's going to minimize competing interactions. We're going to see increasing surface area. Anytime you have infoldings, anytime you have infoldings like this or extensions, you're going to increase surface area. And so one thing you'll often see is this folding concept or these extension type of concept that increases surface area for things to happen. And then in addition to that, these internal membranes, they kind of compartmentalize those processes. They house certain enzymes, and that allows the cell to overall become more efficient. So these are just some examples, okay? When you're doing your cell lab on parts, You're going to be looking at the structures and functions of these different cell parts. Like here's your endoplasmic reticulum. Notice it's a wavy membrane network. Okay, notice it's kind of continuous with the nuclear envelope, that membrane that surrounds the nucleus. We also have these vesicles that transport. Here's our Golgi, our packaging processing center. So notice these extensions and foldings, and all of that is about increasing surface area for chemical reactions to take place. These are all part of what we call the endomembrane system. So you have the nuclear envelope, you have the endoplasmic reticulum, you have the Golgi bodies, and they all kind of create this internal membrane system. Here's a couple other examples. These are your mitochondria and chloroplasts. Remember, these are your evidence for the endosymbiotic theory. Notice the foldings, notice the stacked membranes here. That's all about increasing surface area. And so these guys were those double membrane. They have their own DNA, their own ribosomes. So these are both really special and complex little organelles. And that is going to wrap up our cell notes. So once again, you guys are going to have a quiz here in a few days. Your quiz is going to be over this set of notes as well as the cell lab that you're doing for the next couple of days. One other thing that you're going to get is today you're going to get a bonus opportunity. And I just wanted to take a minute to explain how that is going to work. So each 9 weeks you will get a bonus opportunity. It is a 10-point test bonus that you do not have to do, but I highly recommend it. It is always meant to be homework. And so for this particular bonus, you're going to need to take it home, you're going to need to finish it tonight, and you're going to turn it in tomorrow, first thing when you walk in the door. So the bonus is normally really pretty simple. For today, you have a list of names. All of those people in some way contributed to what we know about cells. And so your job is to research them and write out how do they contribute to what we know about cells. Now keep in mind that scientists sometimes have different hats and they do different things, so be sure you're focusing on the cellular component. And so you just have to take it home, you have to do it, and you have to turn it in. And that will earn you 10 points on a test. Now that's kind of a big deal because this 9 weeks you're only going to have 2 tests in the grade book, and so that 10 points can make a big difference. That 10 points will also be the bumping criteria. So if you do not take advantage of the bonus and your grade ends up with a 9, a 69, 79, 89, then what you're going to see on your report card is a 9. Okay, so once again, the bonus is a bumping criteria. But if you take the time to do the bonus and you put in a little extra effort, then if at the end of the 9 weeks you end up with a 9, that bonus could be worth a little bit more than 10 points, and some points could be added to bump you to the next grade. So just make sure you take advantage of this opportunity, especially after if you weren't feeling too confident after the biochemistry test. So just take that home, get that complete, and turn it in tomorrow. All right, that's all I have for you today. I hope you guys have a wonderful day. You are going to be getting your study guides, and just remember your study guides are a resource to help you, and it's also going to include your essay for the unit, which will be over our next set of notes. So we'll talk about that at a later time. Have a great one.